Hello and welcome to today's webinar on Mastering IHC Staining Experiments. Your host will be Judith Langenig, one of our product managers here at ABD Ceratec. Judith received both her BSc and PhD degrees in molecular biology from the University of Dundee, Scotland. Her thesis research in the group of Professor Jeffrey Williams focused on elucidating STAT signaling pathways in the social amoeba Dictyostelium discoideum. For her postdoctoral research, Judith moved to Dr. Ketan J. Patel's group at the MRC Laboratory of Molecular Biology in Cambridge, where she investigated the role of the Fanconi anemia DNA repair pathway. Since leaving academic research, Judith has held product management roles in both Germany and the United Kingdom. As we're trying to make this webinar as interactive as possible, please submit questions or comments via the Q&A box located at the bottom of the screen throughout the webinar. We will try to answer as many questions as possible in the session following the presentation. However, should any questions remain unanswered, we will email you a response. The webinar will be recorded, and an on-demand version will be available shortly after the webinar on the ABD Ceratec website. Should you encounter any technical problems during the webinar, please click on the question mark at the bottom of the screen. This will bring up a technical guide which describes solutions for the most common technical problems. I hope you enjoy the webinar, and I now hand over to Judith.
Hello everybody. So what topics will we be covering in today's webinar? There will be a quick introduction into IHC. What is it and what is the actual definition? From there, we will be moving on to sample preparation and all the steps that fall into that category. The sample preparation section will be followed by the staining part of the IHC protocol. And from there, we will be moving on to the troubleshooting part, and finally the Q&A session. What is immunohistochemistry? The word histo comes from the Greek term histos, meaning tissue. In an immunohistochemistry or IHC experiment, a primary antibody binds specifically to a protein of interest present in a tissue. The antibody binding is then visualized by a detection system, which provides information about if and where the protein is present in the tissue. IHC is a common method in diagnostics to determine morphological abnormalities and the presence of biomarkers indicative of certain diseases such as cancer. An example of an IHC staining of proliferating epithelial cells in villi with rabbit anti-MCM2 and mouse anti-cytokeratin 18 antibodies is shown here on the left. As the terms immunohistochemistry, immunofluorescence, and immunocytochemistry are often used interchangeably, I thought I'd quickly define the terms according to the guidelines we are using on our website. We only refer to an application as IHC when tissue is being stained. When staining cells, such as healers or gel cuts, we refer to the application as immunocytochemistry or ICC. This term originates from the Greek word cyto, meaning cell. As immunocytochemistry is performed with fluorescent labels, you will often see the expression IF-ICC. IF in this case stands for immunofluorescence. To distinguish the detection labels used in IHC experiments, we differentiate between fluorescent IHC, shown here on the left-hand image, and chromogenic IHC, shown here on the right-hand image. IHC protocols can be separated into two main parts: sample preparation and staining. Sample preparation can be further divided into 4 steps: fixation, embedding or snap freezing, depending on if you're performing IHC paraffin or IHC frozen, sectioning, and antigen retrieval. We will start by focusing on the first step, fixation. Fixation is required to maintain and preserve tissue morphology. It stops decay or decomposition, which is triggered by enzymes present in cells or tissue that destruct cellular structures, a process known as autolysis. It also stops putrefaction, a process caused by bacteria and fungi that breaks down proteins and eventually the cohesion between tissues. 2 main types of fixatives are used in the laboratory, and I'm sure many of you have come across these. The first type are cross-linking reagents such as formaldehyde, glutaraldehyde, and paraformaldehyde. And the second type are protein denaturing agents such as ethanol, methanol, and acetone. The morphology of certain structures is best maintained with a dedicated fixative, such as picric acid in the case of glycogen. But why does the fixative type and the sample preparation method matter? Can't I always follow the same protocol? The sample preparation matters, as your IHC staining result will only ever be as good as your sample. Therefore, care has to be taken in order to identify the best method for staining your proteins of interest. Sample preparation techniques, fixation methods, and antibody suitability are also closely linked. As we will be discussing later, not every antibody is suitable for every of the three main sample preparation methods, which are IHC paraffin, which is performed on paraffin-embedded tissue sections, IHC frozen, which is performed on frozen tissue sections, IHC free-floating, which is performed on free-floating tissue sections, and a method commonly used in neuroscience. In this webinar, we will be discussing IHC paraffin and IHC frozen. Due to time constraints, I'm afraid we will not be able to address IHC free-floating. Depending on which IHC method you will be using, you will either fix the sample and embed it, as is the case for IHC paraffin, or you will snap freeze the sample and fix afterwards. I will first talk you through the IHC paraffin method and then move on to IHC frozen. As we have discussed fixation already, I will discuss fixation in the context of an IHC paraffin protocol and focus on the IHC paraffin embedding step, which gives the method its name. IHC paraffin is the most common IHC specimen preparation. Fixation is required as soon as the sample is extracted and is normally performed for around 6 to 24 hours. Fixation is followed by rehydration in a series of alcohol solutions. After that, a process known as clearing is performed. The purpose of clearing is to remove the alcohol via immersion in xylene solutions. Without the clearing step, the tissue could not be infiltrated with the paraffin wax, as alcohol and wax do not mix. Xylene is therefore used as an intermediary agent, as it mixes with both the wax and the alcohol. After the immersion step, xylene will have displaced the alcohol, allowing the tissue to be infiltrated with wax. Xylene and other clearing agents such as toluene or chloroform are highly toxic. We therefore recommend to always perform clearing in a fume hood. Some laboratories have made attempts at replacing xylene with isopropanol and methyl salicylate. The latter, however, is highly expensive. If you were wondering about the name clearing, the name is due to the tissue becoming transparent once the infiltration is complete. After clearing, the tissue can be infiltrated with hot paraffin wax, which is normally around 60 degrees hot, and a mixture containing other components such as resin. The wax is allowed to cool to 20 degrees in order to solidify. Then the tissue block is cut into sections with the help of a microtome. To summarize this slide, fixation is required to maintain tissue morphology. This step is followed by processing, which comprises dehydration, clearing, and infiltration of the tissue with paraffin wax. Embedding is followed by cutting the tissue into sections. After sectioning, the tissue is mounted onto a slide to which heat is applied in order to improve adherence. The heating step should be as short as possible, as some antigens are highly susceptible to heating. Mounting is followed by deparaffinization and rehydration steps. Paraffin removal is essential for a good staining. Rehydration is performed by immersion in xylene and alcohol solutions. Most commonly, ethanol is used, and the concentration is reduced with each immersion step. Generally, one starts with a 100% solution and moves down to a 70% solution, or in some protocols even a 50% solution. The final step is an immersion in tap water. Ensure that during the entire procedure the slides never dry out, as this could increase background staining. Before the slides can be stained, antigen retrieval has to be performed in order to counteract the fixation or cross-linking step, which has masked the antigens. Two main retrieval methods exist. These are here, heat-induced epitope retrieval, and here, proteolytic-induced epitope retrieval. To summarize the slide, fixation is required to maintain tissue morphology. The step is followed by processing, which comprises dehydrating, clearing, and infiltration of the tissue with paraffin wax. Embedding is followed by cutting the tissue into sections. Sections are then mounted, deparaffinized, and rehydrated, and antigen retrieval is performed. We will now be discussing the two antigen retrieval procedures. Here, which was developed later than PEER, is now the main antigen retrieval technique and performed by heating the sample in buffer solutions. The heat assists with the unfolding process, whilst the buffer preserves the desired protein conformation. Here can be performed in hot water baths, pressure cookers, microwaves, and steamers. If you would like to know more about what device best suits your needs, there's an excellent article called Overview of Heat-Induced Epitope Retrieval Techniques and Devices available on the IHC World website. The article compares all heat devices and their limitations. For example, it highlights that it is hard to use water bath and vegetable steamers, as it's nearly impossible to get them over a temperature of 95 degrees. Microwaves appear to have so-called hot and cold spots and are therefore kind of tricky. All of these limitations have resulted in an increased popularity of pressure cookers, as these can be considered most reliable. When performing here, care should be taken, as here might cause tissue sections to lift off slides. Therefore, always make sure you use tissue adhesive slides, such as polyallysin or APS-coated slides. With regards to buffers, the most common here buffer is a 10 millimolar citrate buffer at pH 6.0. Should you like to make this buffer or another antigen retrieval buffer, please go to abdserotech.com retrieval, where you find detailed protocols. Alternatively, if you are intending to detect an antigen for the first time, you could start with a commercial buffer, such as buffer 25A, which is a neutral buffer. Depending on the experimental outcome, replace the buffer with either a more acidic or basic buffer. In addition to pH, the temperature and duration of the here procedure are also critical parameters and have to be optimized. In order to evaluate the effectiveness of your retrieval method, include a control sample in your experimental design, for which you do not perform here. We will come back to the importance of that control in the troubleshooting section. In contrast to here, PEER uses enzymes such as proteinase K, trypsin, and pepsin for the antigen retrieval procedure. These enzymes degrade the peptides masking the epitope, which has become squashed by the cross-linking process. As PEER might alter the specimen morphology or the antigen itself, it is used less frequently than here, which acts by restoring the secondary and tertiary structures of an epitope. For guidelines as whether to perform here or PEER, please go to our blog and read the post entitled Should I do PEER or here? Also, the Human Protein Atlas might provide useful information as to what type of antigen retrieval and what buffer is most suitable for the detection of your antigen of choice. Often, antibody companies offer specific protocols for the antigen retrieval and detection steps of crucial markers. The example shown here is for the antigen retrieval of F480, a murine macrophage marker. Please feel free to go to abdserotech.com F480 IHC to find out more. You might remember that earlier on in the webinar I mentioned another sample preparation method called IHC frozen. I would like to explain now how this method is performed and how it differs from IHC paraffin. The IHC frozen protocol starts with snap freezing the tissue in liquid nitrogen or dry ice. One needs to be very careful to perform the freezing process as quickly as possible, as slow freezing can result in the formation of ice crystals that can shatter the tissue. Once frozen, the tissue can be cut with a cryostat. The cryostat temperature should be around -20 degrees. If the cryostat is warmer, the section may stick to the knife. A too low temperature, on the other hand, will result in tissue curling. Once cut, the sample can be mounted onto a room temperature warm slide and fixed. If you remember, fixing was the first step in the IHC paraffin protocol. In the IHC frozen protocol, the fixation step is performed after the sectioning. Also for frozen sections, acetone is mainly used. Once fixed, the IHC frozen slides can be stored in a freezer at -20. To summarize, the IHC frozen protocol starts with the snap freezing, followed by sectioning, mounting, and finally fixation. Before we move on to the staining part of the webinar, I would quickly like to compare the two sample preparation methods we discussed. IHC frozen is a quicker method compared to IHC paraffin. This is mainly due to the length of the fixation step, which is around 1 to 2 hours for IHC frozen and up to 24 hours for IHC paraffin. Care should be taken as fixation for more than a day increases the risk of background staining and complicates the antigen retrieval process. For IHC frozen, the fixation step takes place after sectioning, while in the IHC paraffin protocol, fixation is the first step. Also, for IHC frozen, acetone is the fixative of choice, while IHC paraffin tends to use paraformaldehyde. Sectioning of IHC frozen samples is performed with a cryostat, while sectioning of IHC paraffin is performed with a microtome. Should you like to detect phosphoproteins, we recommend to perform IHC frozen. This is due to the fact that the freezing process is rapid, which leads to the conservation of post-translational modifications. Publications further state that phosphatase inhibitors can be added to the fixative and that a short treatment with phosphatase inhibitors can be performed prior to the freezing step. Although snap freezing in liquid nitrogen is rarely available in the clinic, the detection of phosphoproteins is becoming more and more important for cancer diagnostics. For example, when oncologists need to establish what targeted kinase inhibitor therapy to prescribe. With regards to storage, paraffin samples can be kept for multiple years at room temperature. In contrast, frozen samples, when stored at -80 degrees, last for around a year. In general, IHC frozen sections are thicker than IHC paraffin. Increased thickness may result in lower microscopic resolution and less well-defined tissue morphology. Another limitation of IHC frozen is the risk of ice crystal formation, which may omit the detection of subcellular structures. This risk can, however, be mitigated by snap freezing the sample and by letting the sample warm up to -20 degrees prior to cutting. As already mentioned, IHC paraffin is a rather long process due to the duration of the fixation step and the need for antigen retrieval. We have now completed the sample preparation part of the webinar and can move on to the staining part. Should you have any questions about sample preparation, please submit these via the Q&A box at the bottom of the page. In order to avoid high background staining in artifacts, blocking of endogenous peroxidases, phosphatases, and biotin is required. If enzyme activity is not blocked and antibodies labeled to chromogens, such as horseradish peroxidase, are being used, the addition of the chromogenic substrate will result in both the desired reaction with the antibody label and an unwanted reaction with the endogenous enzymes. Especially frozen sections have high residual enzyme activity, which needs to be blocked. In order to block endogenous peroxidase activity, peroxide-based blocking reagents can be used. For example, buffer 17b for frozen sections. For paraffin sections, 3% peroxide is routinely added for 10 minutes. Sodium azide peroxide mixtures or methanol peroxide mixtures are also used. These are, however, not recommended when wanting to stain surface markers. Endogenous alkaline phosphatase activity can be blocked with levamisole, commonly used at 1 to 5 millimolar. When using avidin biotin or streptavidin biotin detection systems, endogenous biotin also has to be blocked. For example, an avidin biotin blocking system such as buffer 16 can be used. In addition to enzymes, non-specific binding sites also need to be blocked. This is a very common step for every antibody-based experiment and also performed in other applications such as Western blotting. As a blocking reagent, use normal serum from the same species as the one in which the secondary antibody was generated in. Block with 10 to 20% normal serum. Make sure to never block with normal serum from the same species as the primary antibody was generated in. This type of blocking serum on the one hand could lead to blocking of reactive species and on the other hand results in higher background staining. If serum is unavailable, use bovine serum albumin, non-fat milk or gelatin. Blocking is followed by incubation with the primary antibody. I mentioned earlier that the fixation method, antigen, antibody choice, and sample preparation method are closely linked. This is due to the fact that not every antibody works in every application. Some antibodies are not suitable for IHC at all, and others are just not recommended for specific IHC applications, such as IHC paraffin. It is therefore important to check on the manufacturer's datasheet that the antibody has been tested or validated in your preferred method. Also, when purchasing antibodies, it is crucial to review the applications and the data provided by the antibody supplier. Many suppliers also sort their antibodies via the different IHC applications, as shown here for ABD Serotech's IHC paraffin antibodies. Alternatives to supplier websites include antibody comparison websites, such as CiteApp, which now lists over 2.5 million antibodies. Monoclonal antibodies are mainly used in IHC paraffin experiments. However, when establishing a new protocol or detecting an uncharacterized antigen for the first time, we recommend starting with a polyclonal antibody, as the antigen retrieval efficiency varies and some epitopes remain inaccessible. Therefore, a polyclonal antibody, which recognizes a multitude of epitopes due to its heterogeneous nature, provides a definite advantage over a monoclonal antibody, which recognizes a single epitope only. That said, if protocols exist, mouse monoclonals or RAB-maps, rabbit monoclonals, are mainly used due to their high specificity. In general, when deciding between a monoclonal and a polyclonal antibody, one needs to balance higher specificity versus sensitivity. Although expensive, it might be worth trying more than one antibody when initially establishing an IHC protocol. A recommendation is also to check the antigen expression levels and localization in the scientific literature, on antibody supplier datasheets, and the Human Protein Atlas. In order to evaluate your experiment, you need to be confident that the antigen is expressed at reasonable levels in your tissue specimen and that you confirm its correct localization. For that purpose, you should always include appropriate controls. Examples of those are autofluorescence or endogenous tissue background staining controls. As certain cell and tissue types, especially those rich in elastin and collagen, display natural fluorescence, it is crucial to observe samples microscopically before every staining experiment. The other type is a positive tissue control, and this means that you include a tissue type that expresses your protein of interest. If you do not see a staining in the positive control, something has gone wrong with your staining protocol. Negative tissue control. Include a tissue type in which your protein of interest is not expressed. Therefore, if you see staining in this type of control, you know that the staining is unspecific. Perform a secondary antibody-only control in order to ensure that the secondary antibody does not unspecifically bind to certain cellular compartments or endogenous immunoglobulins. For this control, you basically follow the same staining protocol without the addition of a primary antibody. Absorption controls indicate that the primary antibody used binds exclusively to the antigen it was raised against. For this control type, you inhibit the staining reaction by absorption of the primary antibody with the purified antigen or immunogen. Isotype control. This type of control is used to ensure that the observed staining is due to the antibody binding the desired antigen and not to some general unspecific binding of the immunoglobulin to the tissue. Isotype controls are only used for controlling monoclonal antibodies. The host species and isotype of the control should therefore match that of the primary antibody. Also, isotype controls should be used at the same concentration as the primary antibody. Many of you will be familiar with the concept of isotype controls from flow cytometry. For more information about IHC controls, please go to abdserotech.com/IHC controls. For detecting highly abundant antigens such as alpha-actin or beta-tubulin, you might consider direct detection with a labeled primary antibody. An example for the direct detection is shown here on the left-hand image. You can either purchase a conjugated antibody or conjugate it yourself with the help of antibody labeling kits, such as our LINX kits. For medium to low abundant antigens, indirect detection is recommended. This can either be done with the help of secondary antibodies, such as shown here on the right-hand image, or other amplification reagents. This recommendation is due to the fact that multiple conjugated secondary antibodies bind to a single primary antibody, thereby leading to amplification of the signal. For very low abundant proteins, we recommend using biotin-related secondary antibodies in combination with conjugated avidin streptavidin. This recommendation is due to the high signal amplification of this method, known as ABC method. This is due to a single avidin molecule being able to simultaneously bind to up to 4 biotin molecules. Whenever you are using a new antibody, the dilution needs to be optimized. As a guideline, start with the datasheet recommendation. For example, if the range is 1 in 100 to 1 in 500, I would consider you do a 1 in 50, a 1 in 100, a 1 in 200, a 1 in 500, and maybe even a 1 in 1000 dilution. The duration of the incubation period and the temperature depend on the antibody affinity. As longer incubation periods and room temperature increase the risk of background staining, as a general guideline, you perform overnight incubations at 4 degrees in either the fridge or a cold room. In order to get the most out of your experiment, you might want to detect multiple antigens simultaneously to determine if they co-localize. This technique is known as multiplexing and is increasing in popularity. As the majority of commercially available antibodies are rabbit polyclonals or mouse monoclonals, 2 antibodies from the same host species may have to be used in multiplex IHC experiments. For example, 2 mouse monoclonal antibodies. This is problematic as the labeled secondary antibodies will bind to both primary antibodies. One solution might be to select isotype and subclass-specific mouse secondary antibodies. For example, for the detection of mouse IgG1 and IgG2b primary antibodies, labeled anti-mouse IgG1 and anti-mouse IgG2b secondary antibodies could be used. Other alternative solutions include the use of conjugated monovalent subfragments that bind to primary antibodies and simultaneously block the binding of other secondary antibodies to the primary. Yet another alternative is to select primary antibodies conjugated to labels that can be detected by other antibody reagents. For example, you can use a FITC conjugated mouse monoclonal antibody and a biotin-labeled mouse monoclonal antibody, which can be detected by anti-FITC antibodies and streptavidin. These secondary reagents can either be directly conjugated themselves or can be detected by tertiary reagents. The primary antibody step is followed by washing steps prior to the incubation with the secondary antibody. When using secondary antibodies, be aware of potential species cross-reactivity of the secondary antibody with endogenous immunoglobulins. If the experiment is poorly designed, the secondary antibody might also react with other primary antibodies than the one that it is intended to bind. As a general rule, use secondary antibodies that were all raised in the same species. Generally speaking, goat and donkey are the most popular species for secondary antibodies. This reduces the risk of species cross-reactivity and enables one simple blocking step with donkey or goat serum. To reduce species cross-reactivity, select cross-adsorbed or pre-adsorbed secondary antibodies. Cross-adsorption is an extra purification step that increases the affinity of an antibody by passing it through a column loaded with serum proteins from a variety of species. When selecting secondary antibodies, you can choose between secondary antibodies conjugated to chromogenic and fluorescent labels. The most popular chromogenic labels are alkaline phosphatase and horseradish peroxidase, while the most popular fluorescent labels belong to new generation dye series such as Alexafluor or Dilate Fluor dyes. To compare the two detection methods, chromogenic labels are compatible with organic and aqueous mounting media, while fluorescent dyes are only compatible with aqueous mounting media. Organic mounting media can often lead to better definition and crisper images. We will talk more about mounting media in the cover slipping and analysis section. Samples stained with fluorescent dyes have the tendency to lose their signal intensity, while those visualized with chromogenic labels can be stored without significant intensity loss. Many kits are available for chromogenic detection, while fluorescent dye series such as Alexafluor dyes are comprehensive and include around 19 dyes, ranging from blue to near-IR emitting fluorophores. This variety of dyes provides many choices for multiplexing experiments and makes it easy to choose dyes that are spectrally separable. Multiplexing experiments with chromogenic labels, on the other hand, are a lot more complicated. When performing a double staining for two proteins that localize in the same subcellular compartment, be aware that once the first chromogenic precipitate, a result of the chromogen and substrate reaction, has stained or saturated the compartment, it becomes very difficult for the second one to stain the same compartment adequately. Designing experiments with multiple enzymatic labels also has its challenges due to the limitation of color detection combinations. Due to time limitations, we will be not able to cover substrate choices in this webinar and move straight on to counterstaining. However, in the troubleshooting section, I will discuss a recent BioCompare benchtip article that provides background information on this topic. Counterstaining provides background contrast and puts the observed staining into perspective, such as defining the plasma membrane and the nucleus. When selecting the optimal counterstain, ensure your counterstain is spectrally differentiable from the color of the precipitate, which is, as mentioned before, the result of the chromogen and substrate reaction. For ease of use, many reagent companies provide mounting media containing counterstains. For more information about the most popular chromogenic nuclear counterstains, please go to our blog article on counterstains. How do I select a fluorescent counterstain? As discussed for chromogenic counterstains, ensure that the counterstain you select is spectrally differentiable from the color of the antibody staining. For example, use the red-emitting drug 5 rather than the blue DAPI when using antibodies conjugated to blue-emitting fluorophores such as Alexa 405 or Dilite 405. For more information about the most common fluorescent counterstains, please go to abdserotech.com/blog/counterstains. Counterstaining is often either simultaneously performed with cover slipping or followed by the cover slipping step. Mounting media are essential for making permanent slides and assist in making the cover slip adhere to the slide. Mounting has the purpose of protecting the specimen from damage while adding contrast during microscopy. Two types of mounting media exist: aqueous or hydrophilic mounting media. Examples of aqueous mounting media are glycerol and glycerol jelly. Organic solvent-based or hydrophobic mounting media. Examples of those are Euperol and Canada balsam. Mounting media can be further differentiated into media that solidify or remain liquid. Factors to consider when selecting mounting media. Do not use organic mounting media for fluorescent labels. When using organic mounting media, care should be taken as some precipitates are soluble in the alcohols used during the IHC protocol. One of those examples is the red precipitate formed as a result of the reaction of horseradish peroxidase and the substrate AEC. For wide field microscopy, mounting media that solidify should be used. For 3D imaging, mounting media that stay liquid have to be used. When using mounting media that remain liquid, the cover slip edges must be sealed with nail polish to prevent the medium from leaking. To prevent photobleaching, a process in which the chemical destruction of a fluorophore results in the loss of fluorescence, slides should be stored in the dark. Further bleaching can be prevented by using mounting media containing antifade reagents. However, as not every fluorophore is compatible with every antifade reagent, we recommend checking the manufacturer's datasheet prior to use. We have now reached the end of the main IHC protocol, and as we have time constraints, are unfortunately unable to discuss data analysis. I would, however, like to quickly talk about troubleshooting. Just as a reminder, if you've not already done so, please submit any questions you may have via the Q&A box at the bottom of the screen. What could be the reason if I do get insufficient or no staining at all? Scenario 1. The precipitate could have been dissolved in the alcohol used for the dehydration step. The solution is to change the enzyme chromogenic substrate combination. For example, change the HRP and AEC combination to HRP and DAP. The precipitate formed by the latter combination is insoluble in alcohol. Scenario 2. You could be using the wrong mounting medium type. For example, an organic mounting medium for fluorescent labels or for soluble precipitates. Scenario 3. The antigen retrieval procedure might have been suboptimal, and the antigen you were trying to detect remained masked. You can change the pH, temperature, or duration in order to improve the antigen retrieval process. Also, check the negative here peer control to see if there's a difference between having performed the procedure or not. Scenario 4. You might have been using an inappropriate detection system. For example, you could have been using a direct detection system and the antigen you were trying to detect was not abundant enough for this type of detection system. Therefore, check the positive control tissue in order to see if you could detect the antigen in that control type. Scenario 5. The counterstain might have been too strong and has obstructed the detection of the immunostaining. The solution is to reduce the concentration or amount of the counterstain, or even change the counterstain itself. For example, you could consider changing it to a different color. What could be the reason if I get too high background staining? Scenario 1 could be that there is too much remaining endogenous enzyme activity in the tissue, and therefore ensure that you're blocking sufficiently and also make sure that you're blocking all enzyme types. That includes the peroxidases, the phosphatases, and also the endogenous biotin if you're using other than biotin-based detection systems. Scenario 2. The blocking step might have been too short or performed with the wrong serum. Make sure to use serum from the same species as the secondary antibody was raised in. And this is really, really crucial for every IHC staining experiment. Scenario 3. You might be using too much primary antibody or too much secondary antibody, or the dilution is indeed too low. So re-optimize the amount and perform a dilution series. You should also keep in mind that especially for polyclonals, each antibody batch needs re-optimization. You cannot just use the same amount or dilution that you were using for the previous batch. You really need to re-optimize each individual batch. Scenario 4. The secondary antibody might be cross-reacting with endogenous immunoglobulins present in the tissue. This is especially a problem if you use tissues such as spleen. The solution is to use cross-adsorbed or pre-absorbed secondary antibodies. Scenario 5. The incubation period with chromogenic substrate could have been too long, or you could be using too much substrate. The solution is to reduce the incubation duration and also consider altering the substrate amount. Scenario 6. You could be washing inadequately. So this could either be that you're washing for too short a period, you're not doing enough washing steps, or you're indeed using the wrong buffer type for washing. So these are all factors to troubleshoot. And then scenario 7, check controls for clues. And this is really, really important. And I recommend that you think about the controls at the experimental design step. So for every experiment, you can only be sure that what you're seeing is real and you're not looking at an artifact if you're indeed checking your controls. So when you're checking your controls, what should you be looking out for? Does the tissue itself autofluoresce? Does the secondary antibody only control show a signal? Then it's most likely that the secondary antibody cross-reacts with endogenous immunoglobulins. If your negative control shows background, the reason could be that the blocking serum was contaminated. If you have the suspicion, leave a small amount of serum in a vial at room temperature overnight on your bench and check for growth the next day. You could also move the serum to 37 degrees if you wanted to. You can spot a contamination by the serum getting cloudy, and this is normally a sign of bacterial growth. In addition to the serum, it could also be the case that your antibody is indeed contaminated. What happens if I can't distinguish my staining signals? Bleed-through of one fluorophore into another fluorophore channel might be occurring, and this complicates the observation of discrete signals and the evaluation of especially colocalization experiments. To avoid bleed-through, select fluorophores that have no or minimal spectral overlap. This can be done with the help of so-called spectrum viewers that are available on the websites of many dye and microscope manufacturers. These tools indeed allow you to mock up the excitation and emission spectra and thereby enable you to select the best fluorophore combinations. quantum dots, which have broad absorption and very narrow emission spectra, are also perfectly suited for multicolor experiments, and we recommend that you actually look at those, and especially if you're trying to do experiments with 4 and more colors, they are a really good choice in order to get spectral separation and a good kind of amount of fluorophores. For more information on the topic, feel free to consult our 10 tips for selecting and using fluorophores in IF experiments website or download the PDF. We also recently published a BioCompare benchtip article on selecting fluorescent and enzymatic labels for multicolor IHC experiments that provides you with hands-on tips. This article also covers the topic of chromogenic substrate selection for multicolor experiments and highlights the most common combinations for which we unfortunately had no time in this webinar. Should you like more information about these articles or would like us to mail you the respective PDFs, please let us know via the Q&A box. We also have a variety of readily available IHC protocols and resources that can be downloaded from the application section of our website. Many of the tips I mentioned can also be found in our IHC Tips and Tricks ebook, which can be downloaded at abdserotech.com, Applications. I would like to let you know that we will be moving on to the Q&A part of the webinar. Thanks for all of you who have submitted questions and have provided feedback. I have seen that a few of you sadly already had to leave us. Just in case you are not able to stay for the duration of the Q&A session or missed your chance to submit a question, you can contact us in one of our 3 offices in Oxford, the United Kingdom, Raleigh in the United States, and Puchheim in Germany. We offer technical support in English, French, and German, and you can simply call us or send us an email. For North America, please email us at abdtechus@bio-rad.com. For the United Kingdom, you can contact us via abdtechuk. And for Germany, you can contact us via abdtech.de. Also, if you just like to find out more about our technical resources in general, just go to abdserotech.com/technical. Alternatively, you can meet us in person at a conference near you. We will be exhibiting at the World Veterinary Vaccine Conference in Madrid in November. Just stop by booth number 10. Another opportunity to meet us is at the American Society of Cell Biology Conference in San Diego, and that will be taking place from December 12 to 16. And to see us there, please stop by booth number 536 and pick up your free kind of copies of our IHC resources. I'd also like to let you know that I've actually enlisted some support for this Q&A session. So I'm actually currently joined by Dr. Tom Augustin. And Tom is one of our technical support specialists. And he's actually got, for his PhD research, he was working in genetics of breast cancer and ovarian cancer at the University of Aberdeen in Scotland. And during his time, he actually performed a couple of IHC staining experiments. That's why Tom is joining me this evening here in the UK. And he's got experimental experience with snap-frozen tumor biopsies, and these he received from the Aberdeen Royal Infirmary, and he was actually looking at a correlation of his samples and BRCA mutation. So I'll be taking a couple of questions, and then I'll hand over to Tom. So the first question we received here is from Krista, and I'd like to answer this question because this is something that many of you will have come across. And this question is looking into mouse-on-mouse staining. And I know Rajesh asked a similar question about rat-on-rat staining. So I thought since this seems to be a common topic, I'll address this one. So what Krista asked us is she's using mouse tissues and wants to, or rather has to, because there's nothing else available, use a mouse monoclonal antibody for detecting her antigen of choice. The protein she is looking to detect is of rather low abundance, and therefore she can't use direct detection with chromogenic or fluorescently labeled primary antibodies. So what to do? So even though you're saying you can't do direct detection, Krista, you might remember in the multiplexing part of the webinar, you were talking about a trick using biotinylated primary antibodies. And you could actually just either buy a biotinylated antibody if that's commercially available or use a kit to label it. And then you could just detect this antibody with conjugated avidin or strapped avidin. And that could be conjugated to a fluorophore or an enzyme, depending on what you would like to do. So this is like the suggestion how you can actually circumvent this problem of detection on mouse and mouse. But I'd like to elucidate this question a bit further because I think it's such an important topic and many of you will actually come across this. So Krista, if you wanted to use a secondary antibody-based detection method, your experimental setup will be problematic, but there are some kind of tips that we can give you to maybe improve this. So the dilemma you're facing when using a secondary antibody is that the anti-mouse secondary antibody will not only bind to the mouse primary antibody, but also to endogenous mouse immunoglobulins present in the tissue. And this is quite a big problem. And in order to avoid this problem and to mitigate you getting false positive results and backward staining, you need to develop a mouse-on-mouse staining strategy. And there is no solution that works for everything or no size fits it all. Some people even report if they're doing mouse-on-mouse that they don't see any background, they don't have to do anything special. So it really depends on what you're doing, how abundant your antigen is. But one way, and that's something we are recommending here, is you could actually look into buying a kit. So there are some actually mouse-on-mouse kits available that you can buy to solve this problem. Or you could actually use so-called Fab fragment antibodies. And we briefly covered them in the multiplexing part. But I just want to go a bit further into what you can do with this Fab fragment antibodies. So you can actually use unlabeled anti-mouse Fab fragment antibodies. In this case, because you are looking for a mouse monoclonal antibody on mouse tissue. And if you're using these antibodies, they should be actually raised in or these fragments rather, they should be raised in the same species as the secondary antibody was raised in. So in your case, Krista, you were thinking of using a secondary antibody, maybe something that was raised in goat. You could be using a goat anti-Fab fragment. The Fab fragments are also used to block binding sites on the mouse tissue. And since all other binding sites on the mouse tissue will be blocked because you're using the Fab to actually do the blocking, your secondary antibody has no other choice but to simply bind to the primary antibody. So in this way, by using the Fab to block all the endogenous immunoglobulin sites, you're forcing the secondary antibody to bind to your primary antibody. And if you're sort of wondering how much Fab to use, so we are recommending between 20 to 40 microgram. And also some protocols recommend you're actually doing a mild cross-linking step of glutaraldehyde in order to reduce the risk of the secondary antibody actually displacing the Fab. So just one thing to keep in mind, Krista, is if you're using the Fab fragments to block, you might have to use more primary antibody. I will be handing over to Tom now, who will be taking the next questions.
Hello, yes. The second question I have here is from Andrea, and they're asking, does fixation of frozen samples affect surface markers such as CD3, CD4, CD8, and do I always have to fix frozen samples? In answer to the second part of your question, the general answer is yes, you need to fix. Fixation prevents loss of antigen and maintains cellular structure. It also permeabilizes your tissue and cells to allow antibody penetration. So you would need to fix. Now, whether your fixation affects surface antigens such as CD3, CD4, and 8, that really depends on the fixative you're using. Fixatives such as glutaraldehyde and formaldehyde tend to cross-link and therefore may mask the epitopes your antibodies recognize. However, other fixatives like acetone or methanol do not tend to cross-link the proteins and therefore generally leave these surface antigens in their more native states. So if you are worried about cross-linking or damage to your epitopes of the CD3, CD4, CD8, you may wish to try acetone or methanol fixation rather than formaldehyde or paraformaldehyde fixation. I'll pass you back now to Judith for more questions.
So one thing we've been asked by a lot of you is actually the resources that have been mentioned throughout the webinar. Where can you find them? And there's actually a box on your screen which says IHC resources, and all the information we've provided, the links to the blog posts and also the articles, you can actually click on these links and you can actually download from there or review this. So just have a look there if that was one of your questions. The next question I'd like to take is from Bianca, and she has been asking us about sequential and simultaneous antibody staining. So that's something that you come across when you're doing multiplexing experiments. And I thought since we only briefly talked about multiplexing, I'll take this question. So basically the difference between sequential and simultaneous staining is that in a sequential staining protocol, you're detecting one antigen with one antibody, and then that detection is succeeded by another antigen that's detected with another antibody. While in a simultaneous staining protocol, like the name says, you are basically detecting multiple antigens concurrently. And you're doing this by actually mixing several antibodies together and adding them at the same time. So sequential staining is actually rare compared to simultaneous staining. And this is actually commonly performed when antigens that require different antigen retrieval techniques are being detected. So in order to know whether you actually need to do sequential staining, we recommend you are checking the literature and finding out in advance what's the retrieval procedure, what has been done before, what's recommended, what works best. I'm just handing over back to Tom, who's answering the next question we have received.
Okay, the next question we received is from Carolina, and the question is, what is autofluorescence? Autofluorescence is generally a property of either your tissue, your fixative, or your counterstain that essentially causes it to emit light when excited at a certain wavelength. Autofluorescence is obviously a problem when you are trying to do immunofluorescence or immunofluorescence staining. In most cases, a lot of tissues tend to fluoresce in the green wavelength, which causes it to mask staining or interfere with the staining of FTSE-based conjugates. So normally that is what you would need to look for. Normally you would need to check if your tissue or any of your reagents in your system fluoresce in the same wavelength as your detection antibody, and you would normally either switch to a different detection antibody or quench the autofluorescence through several means, and I can expound on that later when I email you directly that. Okay. And the third question that I have here is from Abu. Yes. And they're asking which is better for HER staining. Is it IHC frozen or paraffin? Well, because I've worked primarily with frozen, I'm more biased towards frozen. However, I would say that in general, frozen tissues are more accessible, are generally fresher than paraffin sections. So generally, the antigen or the epitope tends to be present more abundantly because they haven't been destroyed. So I would say if you are thinking of doing HER staining, I would suggest you start with frozen tissue, preferably one that has been either snap frozen and fixed with acetone or methanol, because it would be easier to detect, or probably your antigen HER2 would probably be there'd be more of it in the tissue for you to detect. And then if you're feeling more confident, then you can then switch on to paraffin embedded section. I will now pass you back to Judith for further questions.
Thank you, Tom. We've received a question from Mark, and Mark was asking, he's new to IHC and would like to know whether ABD Seartech recommends a simple kind of starter kit that gets you started. So with regards to IHC kits, we've got one, ABD Seartech offers one HISTA detection systems kit, which is our Star 3000 kit series, and that's actually recommended for human sections. And the kit includes linking and labeling reagents, and the visualization is accomplished by an ultra-sensitive indirect labeling method that utilizes novel polymer labeling technology. Following on from that question, we've got a question from David here, and David has been very attentive with regards to looking at the slides. And his question is concerning overfixation, because he'd noticed that we had the term on one of the slides, but I didn't actually explain that in much detail. So thanks a lot for this question, David. And indeed, you're right. This term was mentioned on the IHC frozen and IHC paraffin slide comparison. And the term is actually quite overused and somewhat old-fashioned in the literature. So if you have a look, Some people say it exists, some people say it doesn't exist, and if you check the internet, it is actually very controversial as to what people are saying about overfixation. So in order to look at this term, let's distinguish between alcohol fixation, which is denaturing, and paraformaldehyde, which is cross-linking. So alcohol fixation does normally not have the antigen retrieval step, as we've learned, and therefore it could be theoretically possible to overfix, and that's something to definitely keep in mind. So whenever you are establishing a fixing protocol, it's good to maybe play around with a couple of time points, or also maybe ask your IHC facility or ask some other colleagues for recommendations. And I think we've received a couple of questions from you about specific fixation conditions for certain types of tissue, and we'll get back to you about that later. But when you are doing IHC paraffin and you're actually fixing in paraformaldehyde, we would actually recommend, and in a lot of cases, the antigen retrieval should be good enough to actually reverse the cross-linking process. However, that said, everything depends on what antigen you are trying to detect, what exactly you're doing. So therefore, again, if you're actually making paraffin blocks or you outsourcing it to somebody else, just ask them what's the kind of ideal fixation condition, how long should I go for, what would be too much. But in general, when you're doing fixing and paraformaldehyde, it's not so much of a risk factor. However, with IHC conditions that use alcohol-based fixation, it can be a problem. And one other thing to keep in mind, we've now talked about overfixation. Underfixation is a much bigger problem, and it can lead to it's more detrimental than overfixation. So we definitely recommend that you basically look out for that, and as I said, have an idea of what kind of time frame you're going for. So unfortunately, we are at the end of our Q&A session. I would actually like to thank all of you for attending this webinar. You've been an absolutely brilliant audience, and you've been very active. We received so many questions. For those of you who have not received an answer yet, we will get back in touch in the next couple of days. And may I wish you all the best for your IHC experiments and your research in general, and goodbye from the United Kingdom.